episode 265 of the Stem Cell Podcast, RNA Glycosylation, with Dr. Ryan Flynn. Hey, everybody. We are Doctors Daylon James and Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge and stem cell research by talking to some of the brightest minds in the field. The Stem Cell Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the podcast, please rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have Dr. Ryan Flynn from Harvard and Boston Children's Hospital. He's on the podcast to talk about his research on RNA and glycans. We've also got our usual roundup of recent highlights and stem cell news. That's coming right up.
But first, we can't wait to see you at ICCR 2024, happening July 10th to 13th in Hamburg. Visit iccr2024.org to register and check out the meetings program. We'll see you in Germany.
Can't wait for that. Closing in on 3 months. Is that right? Well, it's going to be so much fun, and we hope to meet a bunch of you guys out there at the conference every year. That's a real treat for us. But for now, we've got a few stories, 4 stories from Cell Stem Cell. They're hogging all the limelight this episode of the podcast. The first one is from a friend of the show, personal friend of mine, Justin Achita, who's killing it over at the Keck Medical School and University of Southern California. This is a neat story that really, I think, transcends the, ivory towers of medicine and gets out into the real world in terms of impact and relevance and visibility, I think, for the public. So I think there's going to be a lot of lay people paying attention to this one because it's about traumatic brain injury, which is the leading environmental risk factor for neurodegenerative disease. There's about 60 million cases of traumatic brain injury each year. And it's associated with the accumulation of these neurotoxic proteins, tau, TDP43, amyloid beta, all of which have been linked to progressive neurodegenerative disease. Most notably, perhaps, in terms of that lay relation, chronic traumatic encephalopathy, which is famous in recent years as being a sequelae of all the hits to the head of not just football players, but a lot of professional athletes that take hits to the head. And famously, like with the crazy incidents, like they looked post-mortem at 60 brains and found it in 59 or some crazy thing like that. So yes, very visible, front of mind, no pun intended there, in the public. Also, though, amyotrophic lateral sclerosis, which is Justin Achita's main wheelhouse there, and Alzheimer's disease, other dementias, the accumulation of these neurotoxic proteins is implicated in all of them. And the problem with trying to treat these diseases are numerous, but the first of all, it's not clear what the mechanism is. If it's like a neuron intrinsic, or what neuron intrinsic mechanisms downstream of the injury, and what's secondary to that injury. Also, you don't have models that approximate the mechanistic biology there, mostly because we're looking at cell culture, but now we got organoids. So 3 dimensions, it's key to look at these things in 3 dimensions that approximate the physiology and anatomy of brain tissue. And also, we don't know what are the neuronal subtypes that are injured and how. And there's not a way to perform screens to look at this. And all these questions are addressed in this story from the Achita lab, where they developed a really cool model of injury where they used this high intensity focused ultrasound in iPS-derived cortical brain organoids. And that simulates that physical injury. And what they show is that indeed you get the classic hallmarks of the injury, including neuronal death, you get the tau phosphorylation, and egress of TDP43 from the nucleus. Those are all hallmarks of traumatic brain injury in patients and leading up to these neurodegenerative conditions. Also, interestingly, they found where uh, the types of neurons that were getting injured. And in this case, they were the deep layer neurons, and they found pretty unexpectedly that the proteinopathy there involving TDP43 was like one of the first events in the early stage post injury that drove neuronal death. And then, owing to the fact that this allowed the focus is a lot about the genetic basis of disease, they looked at the iPS cells derived from the C9orf72 patients that have the susceptibility for ALS and frontotemporal dementia. And they showed that in those iPS-derived cortical organoids, there was exacerbated TDP43 dysfunction. So again, drawing the link between that genetic basis for disease and showing that it manifests in this new model. And then here, where they went over the top, as they always do in the Acheta lab, they tell the story and then they find the solution or a solution. In this case, they used this genome-wide CRISPR interference screen and identified this mechanosensory channel, KCNJ2, that in the screen, when it was knocked out, they did better, and then they could inhibit this and showed that it potently mitigated the neurodegenerative processes, including in those susceptible backgrounds, so the IPS patients with the C9orf72. So, I mean, a complete story, developing a novel model that has real relevance and visibility, and using it to validate the model in the organoid system, and then identifying candidate targets, showing that they can be perturbed. I mean, who knows whether or not we're going to be inhibiting KCNJ2 post concussion or something. I don't know what that looks like therapeutically, but we were totally in space on traumatic brain injury and even modeling that. So I think this is a big step forward. Congratulations to the Cheetah Lab once again, making a big imprint.
Yeah, absolutely. The Acheta Lab has been extremely productive here just down the road here in LA. A lot of really exciting work when it comes to neurodegenerative disease. I know he's a colleague of Clive Svensson, who's actually the head of the Regenerative Medicine here Institute here at Cedar Sinai. They work in a similar space. Really interesting model. Like what you alluded to, this is an area of study that doesn't have a whole ton of models. Um, I wonder if the Cheetah Lab's gonna be collaborating with the NFL at some point. Maybe not. I don't know. That's a maybe you'll be talking to the Rams here in LA. I mean, there are certain limitations with the study, that's for sure. And I think the big thing for me is this concept of traumatic brain injury. It's such a broad spectrum, right? It's just a range in that TBI, right? In terms of severity to not severe to severe TBI. Right, and I think there's different grades as well, in that subspectrum, and they acknowledge that, right? They actually directly say that their system doesn't incorporate that full range of biophysical forces that are present in human traumatic brain injury, and that's just hard to replicate perfectly, right? Especially in this organoid model. But they also talk about how ischemia isn't really well incorporated here, blood-brain barrier leakage, although that can also potentially be modeled using, say, organ on chip systems, which have been used to model blood-brain barrier leakage. There's other stuff like microglial activation that they can still incorporate here. But hey, there's ways you can differentiate microglia now and perhaps incorporate those into these organoid models too. So, not to say it's a perfect study. It's not. I mean, they are very straight up with saying the limitations of the study. But there's a power in the model, and a power in the model that hasn't really existed before until now, you could say, in this in vitro culture. So I think it's exciting because it gives them something to build on top of. And as for the mechanistic stuff, we'll see what happens with that, with the KCNJ2 part of the story, the inhibition part of the story, especially if they push that down towards more clinically relevant areas. But I think just a great model to build on, if nothing else.
Yeah, agreed. And all these models could be made more complex, more complete. But I mean, as scientists, I think the best thing we can do is deconstruct the problem, right? And this has addressed a very, one module of traumatic brain injury, and I think an elegant and clean model that is so bore some fruit. And as you said, I said, we were kind of coming from nowhere in terms of traumatic brain injury. Perhaps not coincidentally, a former comrade. Well, I'm sure they're still very close friends, but Evangelos Kaskinas at Northwestern, he dropped a story on BioArchive about 2 weeks before this that also incorporated. It was a different way that they did it. They used stretch trauma, but they were also modeling this physical injury on organoids. And I'm sure, either directly from Justin or they both were in review at Cell Stem Cell or something motivated him to drop that on BioArchive. But I'm sure no coincidence there. Both coming from the Eggen lab, both very interested in ALS. And as many things go in science, this kind of convergent thought. We were totally in space when it came to modeling this physical injury, and here we are with two unique models of traumatic brain injury emerging within weeks of each other, and just goes to show that I think is some momentum building toward this, and I don't think maybe not with the NFL. I think that they are knocking on the door at USC and Northwestern to try and leverage some of these models to get some hard science behind the speculation, which is pretty sure to certainty now about the link between hits to the head and CTE. But still a lot of experiments to be done, perhaps, to show that the NFL is making an effort to protect their players. For sure, they are going to fund this work.
Hint, hint, Dr. Achita. Moving on to another cell stem cell story. We actually have a bunch of cell stem cell here today on the Roundup. This is a paper coming from over there in China at the State Key Laboratory of Molecular Developmental Biology in Beijing. Title is Harnessing Developmental Dynamics of Spinal Cord Extracellular Matrix to Improve Regenerative Potential of Spinal Cord Organoids. So this is another organoid story in the realm of neurodegenerative disease and traumatic neurodegenerative disease, right? So we're talking about spinal cord injury, and this has been an area that has been very closely tied to the stem cell field for a long time now. I mean, we have the Geron trials from back in the day and all that. We're not talking about that at all. This is more of a, it's more of a, an early phase, very preclinical study here. But when it comes to spinal cord injury, we know that it's, a significant issue. It's, debilitating, devastating event. But interestingly enough, when it comes to regeneration of the spinal cord, the regenerative capacities are actually quite remarkable at the neonatal stage. And this goes in line with, I think, a lot of different tissue types across the body. I mean, in the heart, for example, if there's been cardiac regeneration studies that have been done in early neonatal mice, and in contrast to adult mice and adult humans, neonatal mice, early developmental mice can regenerate their hearts pretty well. And this is sort of the similar line of study, similar, approach, similar thought that they're taking here in focusing on the spinal cord. All right, so neonatal spinal cord has this really remarkable regenerative capacity compared to adult spinal cord. Unfortunately, doesn't have that capacity after injury. But what they wanted to really hone in on here was the role of the extracellular matrix, or the ECM, in that regenerative process, because it hasn't been really well established. And the downstream thought is, if that ECM is beneficial in that regenerative capacity in the neonatal mice, perhaps you can harness that for the adult regeneration of the spinal cord, right? So here, in this cell stem cell paper, they found that the early developmental spinal cord actually indeed had a higher level of certain extracellular matrix proteins associated with neural development and also axon growth. But namely, importantly here, fewer inhibitory proteoglycans, and that's compared to the adult spinal cord. So that's cool. They've figured out the phenotypic difference between adult and neonatal, and perhaps that extracellular matrix composition is one of the keys to driving that improved regenerative capacity in the neonatal spinal cord. So they decellularized a bunch of spinal cord ECM from neonatal and also adult rabbits in this situation, uh, preserved the differences, and did a bunch of analyses on what's different between those ECM components, right? So the decellularized neonatal spinal cord matrix actually promoted proliferation, migration, differentiation of NPCs, of neuroprogenitor cells, and also actually facilitated the axonal outgrowth. So a bunch of different phenotypes that are showing that neurons grown and progenitors grown on this youthful matrix, a bunch of phenotypes showing that that youthful matrix can actually facilitate the growth of these cells and make them divide, do all the things that makes them happy, right? And in contrast, that's not truly the case in the adult decellularized extracellular matrix from the adult spinal cord. Okay, and then they dove in a little bit more on the mechanism here in terms of because we talk about this a lot, right? Extracellular matrix is a gemisch, right? We talk about how matrigel is great, but it's not very well defined. And so you got to figure out exactly what is the molecule that's potentially causing that improved regenerative capacity in the youthful spinal cord matrix, right? And they found that pleiotrophin and tenascin, or TNC, in the neonatal spinal cord, decellularized spinal cord, they could be the drivers. They could be the contributors to that enhanced proliferation, migration, neuronal differentiation phenotype. All right, which is great. It's great to hone in on that. And also importantly, as sort of an early, like a preclinical model system, right? They showed that this neonatal spinal cord matrix had a superior performance as actually a delivery vehicle for NPCs, neuroprogenitor cells, and also neural organoids in a spinal cord injury in vivo model, that's cool. I mean, that's one, like I said, one of the ancestral goals of the stem cell field was to use neurons, whatever, as a cell therapy after spinal cord injury. Of course, it's failed in a lot of different capacities, but folks are still working on this, okay, even many years later. So ultimately, this is suggesting that the ECM, the extracellular matrix cues from really early developmental stages, might actually be really important to regeneration in the spinal cord. And there's a lot of hope associated with these types of studies. Wouldn't you agree?
100%. I loved your use of that phrase there, ancestral goals. I don't know if you coined that, but that's exactly it. That was like number one. We're like, we're gonna get people out of their chairs. And so I appreciate what you said there, that this is one of the most, you know, the one that was the most laden, imbued with hope, the goal that was the highest in the sky, and the most visible. And there are people still working on it. And I think a lot of progress has been made, but unfortunately, because the endpoint is so tough and borderline miraculous, I think a lot of people might call the effort, collectively, writ large, a not a failure, but one of the great disappointments. But people are still working on it, and to me here, I think the key advance is that there's a lot of intuition behind. We talk about Evangelos. He had a story about a couple years ago elucidating the power of the matrix in driving differentiation and function of these cells. And there have been a lot of stories like that, I think, that are like, oh, we'll do the comp. We'll take the old versus the young matrix, and we'll show that the young is better. And like, yeah, I could have intuited that. And it's nice to see the experiments being done and to have the functional readouts of cell behavior. But here, they're actually identifying the factors. I think that's the key step forward here, is to put your finger on what exactly is driving that enhanced function. I still think there's a long way to go, though. I mean, until we get our Star Trek tricorders, it's going to be tough to, like, I think it's one thing to do like the subtractive analysis, be like, okay, there's this here and not here, but the configuration, the conformation, the concentration, all the details in terms of the distribution of these matrix factors, I think, is going to be a key element to leveraging them in a scalable way. I don't know. Is the idea we're going to go from Matrigel to chopped up rat spinal cord? I don't think that that's going to be a lasting solution. Maybe better and better for specific applications, but we got to be able to synthesize this at some point, and I don't know where to start there.
Eventually, yeah, I think that is the goal of all of these matrices, right? You have the matrigel, and we know it works. It's been great for growing all different sorts of cell types, pluripotent stem cells as well. But you also have defined laminin, all these other factors, and these are commercially available. Part of it comes down to cost, and as a lot of things do, we know matrigel, even if it's not defined, has somewhat of a defined cost, and it's a little bit cheaper in usually than some of those more defined matrices. So there's that. But I actually like your idea of specifically growing neuronal subtypes on this decellularized spinal cord matrix, because maybe for those subtypes of cells, ectodermal neuronal differentiated cells, this is going to be amazing. This is like super matrigel for all neural cell types, maybe. I don't know.
Super Matrigel. Put a trademark on it, homie. Uh, as you said, it comes to the cost. Comes to the cost, and this is an awkward segue, but I'm using it anyway. I mean, one of the most expensive new therapies out there is about CRISPR, right? We talked about hematopoietic therapies, addressing hematological uh, malignancies, or like sickle cell as a great example, but the treatments are up to $1 million, right? And that's because these are early days, right? And the therapies are still being developed, but they take a tremendous amount of effort and a lot of resources. Hopefully, we can scale those out to make them more efficient and cheaper. And in particular, speaking of hematologic malignancies and genetic disease, we've thought about as like the perfect fit for CRISPR therapies. But another major target out there, which I think a lot of people didn't think would be cracked in this way, is HIV. Um, Antiretroviral therapies, they addressed HIV in like a curative way, I would argue, as long as you adhere to the therapy. But nevertheless, HIV/AIDS remains a serious global health burden because there isn't like an actual cure. There's just this chronic treatment. And what no one likes to talk about is that it's thought that as few as half of HIV patients that know they have HIV, at least, are taking their medicine, are suppressed. Um, and in 2020, last time we had stats for this type of thing, there was approaching 700,000 HIV related deaths globally. Now, major hope sprung forth with this Berlin patient, so-called Berlin patient named Timothy Ray Brown, who underwent a bone marrow transplant from a donor that had this natural resistance to HIV because they were homozygous for this CCR5 null allele, right? And that was a quote unquote cure, and it has been replicated in other patients, but it wasn't, a walk in the park. Timothy had severe graft versus host disease, had to have 2 cell transplants from that one donor in order to reconstitute his hematopoietic system. And that kind of highlights the challenges to this quote unquote curative approach. Um, One, it's tough. Bone marrow transplant it's not what you want to do. It's toxic, right? There's the treatment regimen, also the GVHD, and also like in that period post or pre full reconstitution, you're really immunosuppressed. You can't fight. You're going to get sick unless you're extremely careful. So the toxicity is one challenge. There's also very few people that are going to be a karyotype match, or a haplotype match, that are also going to be homozygous for that null allele, right? So the odds are against you right off the bat. And finally, which is not well known, I don't think, is that these CCR null cells, they can resist a specific tropism of HIV, but there's another tropism of HIV, another form of HIV that displays a tropism for the CXCR4 receptor, and that's how it enters the cell. So the CCR5 null patients aren't going to be protected from that other HIV tropism. So, in order to address these many problems, Matthew Porteous's group at Stanford, they developed a kind of 2-for-1 strategy where they use this Cas9 adenoviral-based delivery mechanism to do, as I said, 2-for-1 in autologous hematopoietic stem and progenitor cells that were transplanted. They at once knock out the CCR5 allele and knock in two factors that endow the cell with resistance to the CXCR4 tropic HIV. There's these two factors, this viral fusion inhibitor they call C46, and this TRIM5 that restricts HIV. So they knock those two things in, they knock out the CCR5. They show that those cells can repopulate, maintain multilineage capacity, and the T cells, they're inhibit infection by both HIV viral tropisms to the effect of complete loss of that CCR5 tropic HIV and a 2,000-fold decrease in the CXCR4 tropic replication. I think it's key to emphasize that point. This second tropism, while there's a 2,000-fold decrease, which is not absolute, it's a critical intervention because you can't just knock out CXCR4. The cells won't reconstitute, they won't home, and they won't function. So it's kind of a hack here that they had to perform to protect against that tropism. And here, a 2,000-fold decrease is significant, relevant. Whether or not that's going to be a functional cure in a clinical context in a whole patient, that's a question that remains to be answered. But I think at least here in this 2-for-1 approach, we have come in full circle. We have an efficient means of genetically manipulating autologous hematopoietic stem progenitor cells to the tune of curing AIDS. I mean, talk about a holy grail.
Yeah, yeah, this is a big deal. I think this is a really, really cool proof of concept. It's not perfect and it's definitely complex, as you've alluded to. Anytime you're talking about a knock-in strategy for something like this, you run into problems of efficiency. And they directly say it, right? I mean, they're not sure if the remaining approximate 30% of the stem and progenitor cells without a knock-in would actually act as a reservoir for the replication of that CCX4 tropic virus, right? So the problem is you have to get rid of all of the virus, right? If you don't have a capacity to get rid of all of that virus, then it's going to stay latent. It's going to stick around, and you don't solve the problem, right? But I'm sure this is something that they're looking into. They're going to hope to address this in the future. And it's, like I said, a cool proof of concept coming from really a world leader in this area, Matt Porteous. I mean, ever since I was at Stanford, I've known him to be this guy, this pioneer in using genome editing approaches for modifying and correcting and alleviating diseases of the blood. That's the ultimate goal for his work. And Cas9 was used here, and Cas9, as we know, is highly imperfect. It's more like taking a shotgun to the genome as opposed to a pair of molecular scissors. So the next version of this is inevitably going to be using some of these more precise editing modalities. And again, I'm sure they're thinking about this stuff. This is, I'm not the expert here. Dr. Porteous is definitely the expert here. And I think they're definitely thinking about this.
Oh, for sure. I mean, a lot of these studies are ongoing in the next years. This is among the first of many, many studies that are starting to be published using variable techniques toward similar ends. Just in the same, I don't know, week or two at Cell Stem Cell, there was a paper from Cynthia Dunbar's group, who's at the NHLBI, and they were studying doing a comp of lenti versus CRISPR delivery in primates, right? So like really a clinical test. And what they found was, I wouldn't say shocking, but I think it shows how cautiously we have to move forward. What they found is that after 2 months, the lenti was like 100-fold higher levels of cells that were retained in the system versus the CRISPR mediated, and the lenti was like broadly polyclonal, all a diversity of clones, where the CRISPR mediated had much less clonal diversity and was like oligoclonal, approaching a level of like clonal hematopoiesis of indeterminate potential, right? So, like, yeah, two studies coming out in the same month that at once show the promise and all the, kind of newfangled techniques and two for one stuff you can do with CRISPR, but at the same time showing that, yeah, we have a long way to go until we demonstrate that these approaches are clinically safe, effective, and whether or not, fundamentally they're preferred over just antiretroviral therapy, which seems to be working for most patients. I don't know how many people would be psyched to undergo autologous hematopoietic stem cell genetic modification and transplant in order to get to a similar endpoint. So, yeah, a lot of work to be done, but as a proof of principle and something that can be utilized toward other, maladies apart from HIV/AIDS, I think this is a home run.
Yeah, absolutely. And we'll see where this goes. Funny side note, I guess maybe not funny, but side note, we had David Liu here for our annual research day at Cedar Sinai. David Liu of genome editing fame. He's one of the labs that's really pioneered the prime editing, base editing sort of approach, these editing modalities for in vitro and in vivo editing. And somebody asked the question, well, is there any reason to still use Cas9 for homology directed repair and a knock-in strategy? And basically he said no. There's really no reason for that because it's so, as we know, it's so imprecise, it's so unclean. Of course his lab is pioneering some of these next generation clean knock-in approaches. And Cas9 was the first wave, right? 10 years ago, when we were all learning how to do CRISPR first, we all stuck with Cas9, and sort of it's become indoctrinated in the verbiage and the vocabulary of CRISPR. It's CRISPR-Cas9, but it's not. There's so many other newfangled endonucleases out there to do these kind of things. So definitely agree with you. We'll have to stay tuned, and as these technologies inevitably continue to evolve, right? So wrapping things up with another cell stem cell paper here. I think we're 4 for 4. This is also coming from Beijing and that State Key Laboratory of Cell Biology. Actually, sorry, this is in Shanghai. My bad. This is a paper titled "Preclinical Safety and Efficacy of Encapsulated Proliferating Human Hepatocyte Organoids in Treating Liver Failure." This is cool because this is actually a clinical and translational report from Cell Stem Cell. And shout out to Sheila Chari. I mean, we're big fans of how they've incorporated this area and this type of article into the journal. I think this is great. And it's also just a reflection of how rapidly the stem cell field is moving towards that clinical and translational element, right? So this is a cool article. The focus here is again on human hepatocyte organoids in treating liver failure, and building on some existing work that's come out of this particular group. They actually showed that potentially by encapsulating hepatocytes in alginate, this biocompatible material, this could be a promising strategy to treat liver failure. As we know, it's a tremendous medical problem, right? But the issue here is getting enough hepatocytes, as it often is for these kind of studies. Primary cells have a bunch of limitations, as we've talked about in the past. I'm a cardiac biologist. I know the limitations of using primary cardiomyocytes and primary neurons and primary anything. Primary hepatocytes do divide to some capacity, but with all primary cells, they have a limit in terms of their upper limit, in terms of how much they can actually divide, right? So, and quality is the other problem in terms of controlling primary hepatocyte quality for actually use for treating liver failure, right? Uh, like I said, this is building on reported work from this lab. They actually previously reported these proliferating human hepatocytes, or proli-HHs, I guess. Not the greatest acronym or abbreviation, whatever. Um, and these proli-HHs, I'm just going to call them proliferating human hepatocytes, they're actually produced in these alginate encapsulation systems, right? They're encapsulated in alginate, mass engineered as liver organoids to improve their overall maturity, which is great. Um, and these encapsulated liver organoids, or ELOs, I like the abbreviation a little bit better there, were inserted intraperitoneally into liver failure animals. Okay, so these are rodent models to actually treat liver failure, right? Which is great. And ultimately, that's what they showed in some capacity, that these ELOs, these enhanced encapsulated liver organoids with these hyperproliferative human hepatocytes, yeah, they did a pretty good job in treating different models of liver failure. That's the other important thing here. So the treatment improved the survival of mice with post-hepatectomy liver failure, um, hyperammonemia and hypoglycemia as well. So again, properly regulating the function of the liver in multiple models of liver failure. That's really powerful here. It also the treatment with these enhanced liver organoids, these encapsulated liver organoids, also protected the gut from permeability in this situation of these post hepatectomy liver failure, normalized serum endotoxin, inflammatory response, and ultimately improve liver regeneration. That's awesome. And finally, if they didn't have enough models already built into the study, they added a drug toxicity model. As we know, acetaminophen can induce liver failure if you take a massive amount of this drug, or even just a relatively high amount. It can cause liver failure in different people, especially those in vulnerable populations like kids. And they showed that treatment and addition of these ELOs, these encapsulated liver organoids, can actually alleviate that acetaminophen induced liver failure. So that's great. And then they wrap things up with a bunch of toxicity, biodistribution studies, ultimately demonstrating that these ELOs didn't have an adverse effect on the animals that received them. And also very important when looking at any sort of super proliferative cell type like these hepatocytes, they're non-tumorigenic. Really great preclinical study in my mind here. You're looking at multiple models of liver failure and how these proliferating human hepatocytes, or encapsulated liver organoids, can actually alleviate those models across the board, right? So they can restore proper liver function in all sorts of models of liver failure, and including a drug toxicity model. So this is really promising, and I think the next step is just to progress towards clinical trials.
You said it. Clinical trials. I mean, we got our guy just walked out of the hospital with a pig kidney, right? So why wouldn't we be doing some compassionate use for these proly ha-has, or whatever they want to call them? I think that the evidence in this paper is sufficient to suggest that we're ready, because the key element here is this isn't induced pluripotent stem cell derived. These aren't liver-like cells. These are legit primary hepatocytes. Here they've demonstrated function. I don't see, I mean, yes, there's some safety stuff to figure out, not least of which is, I always have this question, is if you proliferate something mega outside of the body, what about that whole clonality element in introduction of oncogenesis? I don't know. I think that could be scary. But again, we're talking about compassionate use patients. If pig kidneys can be applied toward this end with living, patients are going to go on, continue their life. It looks like the results are pretty impressive there. I would love to see these cells used in a similar way in patients, and I don't see what the obstacles would be. And as a bridge to more IPS or pluripotent stem cell based therapy, I think that would be so key, because it'd be, I don't want to call it a Trojan horse, because that, we're not trying to fool anybody here. It's just a matter of getting to a point where we're comfortable with cell-based therapies that aren't about the whole organ. I love the idea of these, I hate to use the mini liver or whatever, but these modules that can be replaced and are maybe even kind of disposable or temporary, whatever it is. This is the case use that I'm really looking forward to, Arun.
Yeah, I like how you brought up that concept of a bridge, right? And maybe that's what this is. This is just a bridge until somebody can actually receive a real liver for transplantation, and maybe these cells can buy that patient some time, which is a wonderful thing. Similarly, we have the cardiac side of things, right? Because that's also what they're trying to do when it comes to introducing, iPS derived cells, for example, into the heart after myocardial infarction, whatever, to buy that patient some time, potentially until they can actually receive a real human heart. And I like how you brought up the pig kidney, because maybe that's the end goal. And we've talked about this a lot here on the show. Xeno is moving fast, really, really fast. And if we know that there's a huge shortage in human organs, the next best thing, or the better thing, is to just use genetically compatible, immunocompatible pig organs, because there could be an unlimited supply of that. So we might all be out of jobs pretty soon.
Yeah, man. You talk about bringing back the ancestral hopes, right? We all thought it'd be pluripotent stem cells, and here comes, adult stem cells, which we thought were left behind and out of left field, bringing the pig organs. So it's not what we thought, but it's all good news, if you ask me. Moving on to our guest, who's going to tell us the good news in RNA. But before we get there, quick message from STEMCELL Technologies about a new alternate podcast. All right. It's called Lab Coats in Life, and it's a brand new podcast on mentorship in science. And Arun and I are excited to be co-hosting. We recently chatted with Dr. Rekha Iyer from the New York Stem Cell Foundation on diversity and inclusion in STEM. Find this and other episodes wherever you get your podcasts, or at labcoatsinlifepodcast.com. All right, everybody, with us today from Boston Children's Hospital, where he is principal investigator, also assistant professor in the STEM and regenerative biology department at Harvard University, Dr. Ryan Flynn. The Flynn Lab is focused on exploring the interface of two key biomolecules, RNA and glycans, by developing and implementing chemical, biochemical, and genetic tools. Understanding the regulatory mechanisms underlying glycoRNA and the interface of the secretory pathway and nucleic acids will have implications for the basic biology of processes such as stem cells, cancer, and regeneration. And Ryan's going to tell us all about that. Dr. Flynn, thank you so much for joining us on the show today.
Thanks. And yeah, happy to be here today.
Yeah, thanks for joining us, Ryan. You're a relatively new PI here on the scene after this really productive stint at Stanford with Howard Chang, and then a postdoc with Nobel Prize winner Carolyn Bertozzi. We love highlighting new PIs here on the show. And so why don't we actually start things off with an easy one? Tell us about your lab's RNA-centric view of the cell surface. I got that quote from your website, actually. And since we're a stem cell show, stem cell podcast, maybe tell us a little about how these glycans or strings of sugar that can be found on the cell surface can influence stem cell function.
Yeah. Great questions. And it is a thing that we think about most days. I think, well, when I began my training, I'm an RNA biologist by training, an RNA chemist, a chemical biologist. But I really thought a lot about intracellular mechanisms and how do these molecules, RNA, control all these different parts of the cell. And I had really forgotten about the outside of the cell, which is, of course, very important for how cells talk to each other, how they talk to the environment, how pathogens interact and eventually infect our cells. And the outside of the cell and the cell surface is actually the world of glycosylation. It's where the vast majority of carbohydrate polymers, glycans, perform their role. And so complex organizations of cells, stem cell niches, organization. All of these things require and rely on glycobiology as a principle, but for the longest time, I even forgot that the glycans existed as a biomolecule in the cell. And so really what my lab is doing, and we can talk about it later in terms of the way we got here, but what my lab is trying to understand is how and if RNA interfaces with that really rich and important biology outside of the cell. Right? What RNAs get there? What are they doing there? How are they being regulated or controlling the outside of the cell? And does that impact the ability for the immune system to recognize tissues and for cells to self-organize? If you're a developing embryo or if you're a pathogen, do you ever see these RNAs and how are they impacting that activity? So the sort of the general question for us is, does RNA impact extracellular biology? We think it does, of course, and the way we're thinking about that is through this initial work, this initial discovery of a glycosylated RNA that kind of forces you to think about RNA in a place that it wouldn't otherwise be, but around all of these carbohydrate polymers.
Yeah, and that to me is the real revolutionary idea, the breakthrough. And the idea of thinking of RNA outside of its context, it's flimsy or transient molecule that's thought of as kind of the antithesis or antipode of DNA, which it predates evolutionarily, it's thought. And the biology of RNA is super, super deep. I mean, even deeper than my basic biology, high school education would suggest, where it was like brought down to just the three fundamental operators, right? The messenger RNA, the transfer RNA, and the ribosomal RNA. That's kind of most of what you know about RNA until you specialize in it. But, over the last 2-3 decades, maybe there's been an explosion in our understanding and appreciation of all the other kinds of RNA and their functions. And that's not really to mention even these extracellular cell surface RNAs that you're talking about, which is another breakthrough. But just to mention those the myriad internal intracellular RNAs, the link RNAs, the Pee-wee interacting RNAs, the microRNAs, circRNAs, and that's really the tip of the iceberg. In fact, reading about this, I read that it's thought up to 75% of the human genome encodes RNAs of various types, relative to 2% that encodes, the proteins that we all know and love. 75%, I thought that was mostly junk RNA, so you'll have to tell me if that's accurate. But the point being is that RNA far outweighs proteins in terms of the potential, all the variability and diversity. And of course, owing to that tremendous diversity, there's a fleet now of RNA-based therapeutic approaches, antisense oligos, siRNAs, antagamers, aptamers. As someone who's at the leading edge of both basic and clinical study, I would argue, what do you think is the most exciting RNA-based clinical application that's either coming to the fore now or that you see in the near future?
Yeah, I agree with basically all of that. Of course, I have a bias. I think RNA is the most important molecule in a cell, so that's just important to say up front. One thing I teach a class, an undergrad class at Harvard, and I sort of make a joke in the beginning. We learn about an mRNA, and then we learn about non-coding RNAs, and I sort of make a joke that mRNAs are actually the weird RNAs. mRNAs have this strange function with the ability to be translated into a protein. But every RNA is a scaffold. Every RNA adopts a structure. Every RNA can sense things. Every RNA brings together proteins and has all these other activities. And it's sort of, mRNAs have this one additional feature that is funny and weird and makes a protein. So it's perhaps an interesting perspective to think about when you consider this, like you said, this large fleet or suite of RNAs that are in the cell. There's this enormous amount of role. There's an enormous amount of roles that they play, and translating into proteins is only one, and it's a small feature of a small subset of them. So just as a broader thought. But to come back to your therapeutic question, I think RNA in general is interesting and useful from its sort of obvious programmability. I think that what I like thinking about, a little bit of what my lab is thinking about, is there's sort of, I think there's these two parallel tracks. One is the sort of creative methodological or sort of technology design that you can implement with different tools, targeting of Cas proteins, copy paste things into the genome, like you were saying, aptamers, these mixmers and ASOs. There's this enormous, space to explore from a technology perspective on how do you actually manipulate the molecules at a cell, in a tissue. But I think there's this other parallel track that is very obvious, but has been super slow to address, which is delivery. And I actually think that delivery is somewhere where there are much larger gains in the short term, which will open up longer term, accessibility, literal and figurative, to these therapeutic modalities, right? You look at the statistics of even the mRNA therapeutics, of how many mRNA molecules make it from the syringe into the tissue, into the bloodstream, to the target cell, into the endosome, and then into productively the cytosol. And it's a comically small fraction that is delivered effectively. And that process, which may or may not have relevance to RNA biology, but is something that's really important in nucleic acid therapeutics, is something that I think is really important to think about. How do you get productive, efficient trafficking and delivery of whatever it is? And they all matter differently. If you want an ASO, you need nuclear delivery. If you want an mRNA, you probably need cytosolic delivery. And these are actually not the same, right? Of course, you have to get past the nuclear pore and all these things for getting nuclear activity. So it's not a direct answer to your question, but I think I like to split it up in that way, because there's a ton of exciting stuff to think about from the technology perspective of what could you do with the nucleic acid. But if you can't get it anywhere, who cares? And so we can get it to some places, but you look, at least as of a few years ago, Alnylam was able to target many diseases. But when you look at the way they think of physiology, they're a liver-centric company because they can target the liver. So any drug that they can make that can target the liver and affect peripheral to the liver, they could target that disease. But if it is knocked down in the liver and does not have an effect on your target tissue, it was not accessible by the siRNAs they made. So that really controlled the destiny of what those therapeutics were doing. And so I think it's an interesting pair, because they've solved the problem of siRNA, but they don't yet have the solved problem for every other tissue that you want to actually do productive knockdown in.
Yeah, that's a fantastic summary. And I think it's really important to reflect on that. I mean, we've talked about a lot about how this class of molecules, RNAs, can be used as tools, like you're talking about with CRISPR and Cas9 and targeting and all that. But yeah, it's something a lot of basic scientists may not necessarily think about, is that delivery is so critically important to the success of the downstream therapy. So I think it's worth reflecting on that. I mean, a lot of that work is being done in your neck of the woods, right over there in Kendall Square and Greater Boston, Cambridge. So you're really in a great spot for that. Let's actually dive a little deeper into your lab's work, your basic science work on glycoRNA. I mean, this is a previously undescribed glycoconjugate, which consists of RNAs modified with secretory end glycans presented on the cell surface. I mean, you posted this bioRxiv paper recently, which developed this really sensitive, scalable protocol. I mean, we're talking about tool development, right? To detect and characterize native glycoRNAs. So tell us a little bit more about this unique class of molecules like glycoRNAs, what they're all about, and maybe a little bit more about this new protocol to actually detect them.
Yeah, of course, our main interest in the lab is glycoRNAs, and they fit us into this context of cell surface RNA biology that we're thinking more about. But kind of understanding this hybrid polymer of small non-coding RNAs and N-glycans that are produced in the secretory pathway got us to think a lot about these new processes and these new spaces. And when we, there were a series of things that brought us to ask this question. It was very difficult to understand whether or not anyone had asked if RNAs were glycosylated. It was not well characterized if endogenous RNAs ever made it into the ER or Golgi from the cell that they are produced from. And cell surface RNAs were not really a thing that were understood as a prominent activity. And so the combination of these things made me ask questions about glycobiology with the lens of an RNA biologist. And I'm saying it that way because what we ended up using were really specific and well-characterized chemical tools, these metabolic chemical reporters that install click handles into glycans and then looked for RNA that co-purified, right? And that was the initial discovery and the mechanism that we used to make that observation. And with that data, we built out this model that small non-coding RNAs, these are things like tRNAs and yRNAs and SNO and snRNAs, these very abundant, highly conserved, essential RNAs, that they get in some way trafficked to the lumen of the ER and Golgi, get conjugated to an N-glycan and presented on the surface of the cell. And so it was telling us a lot of new things, but a lot of those tools were challenging to use. You know, if you need a metabolic reporter, and this is true if you're labeling RNA, protein, or glycans, the cells need to be alive. So imagine you want to study a patient sample or some primary sample that might be very sensitive to exogenous molecules added. It would be very challenging to add these modified sugars to them. So that was one issue. And we also knew that metabolic reporters, while very specific, are often low efficiency in their incorporation. So if you have 100 sialic acids, which is one of the types of sugars, you might get incorporation only into sialic acid, but maybe 5 of them get labeled. So you subsample or undersample the scope and the bulk of your molecule of interest. So you might tell that it's there, which we could, and we found glycoRNAs that way, but we weren't able to look at the full suite of what was there. So this newer technology that we've been working on for quite a long time was trying to move away to develop a parallel strategy where you could take any sort of an arbitrary RNA sample, whether it was from a patient sample, was it from a HeLa cell or wherever, and label directly in vitro the sialic acids, derivatize the sialic acids, the chemical approach. In this particular case, we used established glycobiology concepts. So you can use oxidation with a periodate tool to cleave the vicinal diols of sialic acid. They're very labile. And you can then do aldehyde derivatization or aldehyde ligation of those, of the aldehyde you create after oxidation to produce some conjugate. And we could put biotin on it, we could put a fluorophore on it or whatever. And what we learned after doing this was that we were missing a lot of the signal. A lot of the glycoRNAs were going unlabeled when we used those metabolic reporters. And so the initial description of this and the characterization of this was sort of just surprising how much more we could see when we labeled the cells that we had, our cancer cells that we were normally working with. And we kind of took it in 2 ways. We thought, well, this would give us access to new material. So we went down the primary cell route and tried to demonstrate that you could actually label glycoRNAs from a clinically relevant sample. We isolated PBMCs, the mononuclear cells from a healthy person, and labeled monocytes, T cells, and B cells, and could actually detect unperturbed glycoRNAs from those cells, from only 100,000 cells. And this is more than 10 or 20-fold increased sensitivity on that side. And I can get into it in a second, but the second thing we did was we tried to address a big concern and a big question that we had, which was structural characterization, right? When we described the glycoRNA initially, we did everything we could to demonstrate that this molecule was a proper hybrid conjugate between an RNA and a glycan. But what we were missing was the chemical or atomic understanding of the atoms that linked the sugar on the glycan to the nucleobase of the RNA. And with this increased sensitivity, it sort of afforded us an opportunity to look at everything that was in our tube and look for that linker. And so this is one of the things that we've been doing, now, and it's sort of the end half of that work that you mentioned.
I'm going to be straight with you, Ryan. I understood a lot of that, and I understood to lesser degrees some of that. And that's because when you got into the chemistry, it got a little too deep for me. But I love the whole answer. You know why? Because I could see, and I'm on a Zoom here, I can see your enthusiasm, your fluency, and all that stuff. But what really came across is that you're totally in space. It's how you started that answer. You were starting from saying no one even knew about it, whether even how to ask the question. And it's why I love this interview, because we're talking to the innovator, right? You came up with all this stuff from kind of deep space, and now you're developing the tools. And what you just described there is how you kind of putter, right? You tinker. You use the insight from your previous life or from a collaborator, and you combine it and create a whole new tool, a whole new way of understanding the thing, right? And it's a big deal. And that's why the discovery of glycoRNAs was such a big deal and got so much press. there was like all the elements of a great science story, a little bit of serendipity, defying the accepted principles of cellular biology. Can you tell us a little bit more about that facet of it? You know, being kind of in this pioneering space where I'm sure there was a lot of naysayers, a lot of shade, a lot of skepticism. How does the sharing and validation of science play out in that context? Is the bar in review, for example, did you think the bar in review of that paper was higher because you were introducing this brand new concept, or was there like real drive from the editor because of the enthusiasm of a new concept? Talk about that a little bit, if you will.
Yeah, yeah. Yeah, it was interesting. I mean, my goal was not to go to Carolyn's lab and find a new molecule. I did have a desire to bridge RNA biology and glycobiology. That was part of my interest. And I really wanted to study glycosylated RNA binding proteins and think about the interactions there and the functions there. And so when I was designing these experiments, it was a series of 5% degree questions that was like, well, what if this, maybe if that, then this, then that, then this, and if it this, and that. And I basically ended up saying like RNA could get a glycan on it, like a secretory glycan on it, and it didn't make any sense, but I developed experimental evidence around this thing. And so I think it only after probably a year of discussing the results with Carolyn and thinking through the data I mean, she kind of more quickly realized that I was saying something insane, perhaps, and that it would be very interesting if it were real. And she would frequently remind me or ask me if it was real in the hallways when she would see me. She'd be like, is this data still real? Are you still getting data that is agreeing with this? But it was only after some time that we sort of appreciated that this would be very surprising. Like my question was just like, is this signal interesting, and what is the parameters of the signal? The initial work, I had a lot of that data very quickly. I think perhaps I collected the data for that paper within a year, and it was very obvious that what we were seeing was robust, but where we were stuck was, there were two big questions, right? Initially, we didn't know what the linker was. So we didn't know that we couldn't have undeniable proof that it was a single molecule. And we also didn't know what it did. And of course, there's a circular logic there, because the more you know, the more you can test. And so if you knew what was building it, you could knock that out and ask, what does it do? And this sort of thing. We didn't know any of that. And so we initially had sent this in, and we got reviews, and the reviews were, I think, very fair. They were curious and critical of this discovery, and they said, you need to show that it's a real molecule. It would be great to have NMR, have a mass spec, a structure, whatever. And I'm an RNA biochemist. Like, what do I know about getting a cryo-EM or a mass spectrometry, spectra, or even what does a spectra mean? These were things I had never learned before, and I had to learn how a mass spec worked. And I had really, really good people in Carolyn's lab, Stacy Mollacker in particular, who has a lab now at Yale, who was just like every day working with me to figure this thing out. And learned a lot, but the initial questions were, what is it and what does it do? And we made a sort of a breakthrough about where it was that changed the imagination, I think, for biologists. Because I think I'm a biologist. Well, I know I'm a biologist, but I think that many biologists are and have to accept some uncertainty about how biology works. But you need something to go on. You need something to imagine what it could do, where it could be active, why is it relevant? And so when we realized and built up a lot of evidence around it being on the cell surface, it provided an imagination for what this new thing could do. We didn't prove definitively that there was a real function, a particular activity or whatever, which we have, I think, some data on now. But it enabled reviewers and editors, I think, for that initial work that was now published in Cell, to imagine, right, that there was this new space that RNA could work in, that there was a new role for this hybrid conjugate. Now it's very interesting because there are lots of opinions about this as I'm learning, and some are more shared with me, and some are less shared with me. But I think even as far, as recent as November, especially, I think before that bioRxiv we put out about the linker, there was a lot of very reasonable skepticism about the observation, because again, we didn't have any direct evidence, no chemical evidence, no mass spec evidence, no genetic evidence of a proper hybrid attachment between an RNA and an N-glycan. And, we did everything we did. We did everything we could. I've probably been working on that linker for the last 6 years, and we've sent material to 15 to 20 different labs across the world. Like everyone that I could get a mailing address for, I sent material to. We would send milligrams and milligrams and milligrams of RNA, small RNAs. That's 1/10 of the bulk of total RNA.
Wow.
I mean, I can't tell you how much trizol and arnazol I've gone through to extract all this stuff. And so it was very much uncertain how we would find this. What would we do? And it became it was sort of a transformative collaboration that I've had now with Ben Garcia and Axe, a postdoc in his lab, who specialize in PTM analysis. And it's very interesting. Briefly, Axe, this main collaborator we have, he did his grad school in a glycomics lab. And he went to Ben Garcia's lab to learn RNA PTM analysis with mass spectrometry. And so the person who helped us find, who made that spectra, was a person who deeply understood glycans and RNAs and how they flew in a mass spectrometer. And it was like only in that combination with, I'll say, some of the newest instrumentation, right, and using what's known as data independent acquisition for mass spectrometry, did we find this. And it really took a lot of deep thinking, a lot of risk on their part to spend time and energy on this. But we ended up finding that there was this linker called ACE3PU, and that's the end part of that bioRxiv. But I think before that, there was, and still now, but there was a very obvious question of, well, this is all great and fine. You see signal on a gel, but really you haven't proven anything. And I understood that. And that was, we were spending a huge amount of effort thinking about this. And now that we have this evidence, we're under review. We'll see how people are going to evaluate that. It gives a lot of confidence to me about how to continue the investigation and expand the investigation. We have genetic tools to delete that mark and investigate its function. We have mass spec ways to isolate material from different tissues, from different species. Ask, where is this seen? Where is this not seen? What are the other properties? Is it only linked to uracil, which is what ACE3PU is on, or is it linked to other things that we found at the mass spectrometer but couldn't validate yet, like wibutacine? These are in our mass spec data sets. So it's been interesting as we develop new tools and new evidence to see who is convinced by different things. And I presented this work the same day it came out on bioRxiv at the Society for Glycobiology meeting. And I met a lot of people after the talk, and they were like, I have to be honest, I didn't believe any of this, but that mass spec is pretty hard to argue with. And I was like, yeah, I mean, that's true and totally understandable, right? It's very reasonable to be skeptical and think through this stuff. So yeah, it's very interesting to see how it evolves. It is a little slow sometimes because we are doing a lot of the tool development. You want to make a measurement and understand the function, but you can't do it until you can measure it and monitor it. But to measure and monitor it, you have to make a tool. So you end up queuing yourself behind a bunch of these sort of serial processes. But in the end, I think our goal is to make the careful measurements that we can, provide extremely detailed methods. I make a very big point with people in my lab and myself to make the methods extremely detailed, completely transparent. I answer emails very quickly whenever anyone has questions. So you can test that out if you want. But if you ever have a question on the methods, like I email back the same day with whatever we can. Because I feel like, we're only as good as the things we see. And unfortunately, my lab will only be able to do a certain amount of work, right? Other people will have to help us understand this biology. And we want it to be accessible and measurable, right? And so that's sort of where we've been where we've been operating now. And I think this year we'll have some more biological insights. But what am I? I'm an RNA biochemist. I don't know. I studied stem cells starting when I was an undergrad, but I'm not an immunologist. I'm not a rheumatologist. I don't know these systems. And I think what will really come out of this field is when domain experts can ask the outstanding questions that are in their field and say, is glycoRNA relevant here or not? It doesn't have to be relevant everywhere, but maybe some places.
I mean, that is just an incredible story. I mean, and honestly, inspirational, I think, to a lot of the trainees out there who are listening to the show. I mean, it's basic science discovery at its best. I mean, you mentioned how certain technologies were critical to making this happen, how certain collaborations were critical to making this happen. And I think it's also just a sign of a great scientist in yourself, and also a great mentor in Carolyn, that in the face of that skepticism from the community, from this established community, you were still fearless to pursue this topic that, like you said, required a lot of imagination to believe and understand. So we'll definitely be tuned in to see what happens with that paper and the studies. So thanks for sharing that. That was awesome.
One thing I would just mention, I think in the moment, perhaps it doesn't seem fearless necessarily, but I think the way that I approach these things is sort of like, do I care about the question or the answer? And for me, what drove me was to understand whether or not the signal was real. And I decided that a yes or a no was equivalently interesting. And I needed to understand very carefully that outcome. And perhaps it was useful to not know how surprising this discovery would be or interesting it would be, because I was just very curious to know this answer. I needed to answer yes or no, could just be a check, true false box in your questions. And I would have been very happy to know either way, right? Either this was a thing that was happening and no one had seen it before, or it never happens, right? And for me, sort of building up that outcome where they were equivalently interesting outcomes, yes or no, helps perhaps ask a more complicated or difficult question that you don't know the outcome to. But that's sort of the way I was approaching it. It was I thought equivalently interesting, yes or no.
Yeah, it's I think a really good lesson for the trainees out there. I mean, we're sometimes so tied to our hypotheses, right? We want them to be right in some ways, but that's not the right way to do science. You have to be unbiased in terms of your approach, and the curiosity is what should drive you, right? Not the success of the hypothesis ultimately. So some food for thought for the trainees out there. And on that path, we're talking about trainees, training, all this kind of stuff. I mean, you also happen to be a physician scientist, having gone through med school at Stanford while getting your PhD. We overlapped for a little bit. I mean, you started your lab pretty much right after finishing up your postdoc without doing the standard medical scientist training path, right? The standard internship, residency, fellowship that a lot of physician scientists take. I mean, given the fundamental nature of the work in your lab, like we're talking about here today, it makes sense. But could you tell us a little bit more about that decision of going through the MD-PhD training without actually ultimately doing the clinical side of it? And maybe for the trainees who are listening here on the show and are thinking about that path of MSTP, MD-PhD, whatever, I mean, tell us a little bit about what that road is actually like and some of the decisions that you have to make along the way.
Yeah, it's a good question. It's something that I had probably spent an inordinate amount of time thinking about while training, much more than I did before the training. But I guess as a very brief history, my mom was a breast cancer surgeon and my grandfather was a general surgeon, and I had sort of seen them work. I knew of my grandfather's work. I had been into the hospital and the OR with my mom, and this seemed like an incredible thing. Since I was very young, I wanted to be a surgeon. And so this was sort of just my principal vision, and I liked science and these sorts of things as I was growing up, and through high school, and went to MIT and got some really formative experience in lab with Phil Sharp, and did only basic science. Very, very, very deep RNA biology at the transcriptional start site of actually embryonic stem cells, and tried to understand cell states there. And at the end of that time, I still was 100% convinced that I wanted to do, I wanted to have an impact on human health. I wanted to be a physician scientist. And so, of course, I learned about MD PhD. I was like, this is fantastic. I get an opportunity to do both of these things that I find very interesting. So through perhaps a non-canonical path, I actually went to Stanford as an MD only, knowing that I was going to do a PhD, and then kind of made my way into the PhD program there in a secondary side, just by sort of brute force. Ended up as an MD PhD at Stanford, and sort of what I realized was I hadn't ever had any direct individual experience as a caregiver in clinical medicine, right? I think when you do science, you actually there are many more opportunities early on to get direct experience being a scientist, which is awesome. In clinical medicine, it's fairly restricted for good reason. And you obviously do a lot of experiences and you try to get volunteer things and all these other things. And I had perhaps some of the best opportunities and access being in the OR when I was young. But you don't really get that experience of you being the clinical point person, right? You being the source of those decisions, the responsibility holder. And so, it took many years to kind of think about this, but as I did the medical training, got experience in the clinic, and also did more research, there were sort of perhaps two things that I was thinking about, or two different avenues. And one was a little bit of like, where did I want to sort of like, I think the way that I was thinking about it was like fractional likelihood of having an impact and pace of impact, sort of, or the way that impact works, right? So I think as a clinician, you have like a measurable daily impact on the people you see, 100%. But you don't usually, as often is the case, you don't usually make the new medicines. You don't usually innovate there. It's more of the system or partnering with scientists and bringing new things in. Often, everything has exceptions, but you're often leveraging tools that exist to deliver the care, and you're doing the care delivery, right? And then as the scientist, you have like a fairly low statistical probability that's not zero, but of a chance of helping perhaps many people over your lifetime. And so you're like near one or near zero. Neither of them are one or zero. And then the time at which you accomplish those things is also like days or decades. And so there were these like very polar opposite sides. And what I learned was that my personality was much better suited for like low statistical success over a very long time. And that was sort of the more scientific side of things. And so I think that was the way I think about it. That's sort of the way I think about it now. But it was very hard to learn that beforehand. Like being able to have an impact on a person's life is very appealing, and make that meaningful outcome for them was very appealing. But it's very hard to see how do you do that in a clinical setting, whether it's an inpatient, outpatient, whatever blend there is. Can you have a lab? What type of lab? What type of specialty? These things. It's super personal. And so I talked to this number of people about this in different contexts. It's very hard actually to predict, I think, what is your personality when it interfaces with these different verticals, the sectors that make up science or medicine, or a blend between that. But that's sort of how I think about it.
No, I think that makes a lot of sense, and reflects a lot of wisdom on your part to have these internal conversations at a young age, right? That's a big decision to make, because if you go all in on that clinical training path, as you know, and as I know, from my spouse, who's also a cardiologist herself, it's a long road, very,
Yeah.
very long road. And you might not actually start your basic science career until a decade after finishing that road, right? So I think I think you made the right decision ultimately. Things require collaboration, as we're talking about here in the show. MD, PhD, MDs, PhDs, collaboration between clinicians, scientists that ultimately bring these therapeutics to people and to make these discoveries happen. And I think you have a an amazing collaboration in your personal life as well. I mean, we had the good fortune of having your other half, Dr. Ritu Raman, here on the show not too long ago. Congratulations on your marriage, by the way.
Thank you. Thank you.
Yeah, I mean, she mentioned how you guys are navigating that journey of new PI-hood together in Boston. And tell us your half of the story. I mean, we chatted with her. What's it like getting started in your independent career in Boston with a partner in science with whom you can share everything?
Yeah, well, I think it's been great so far. You know, I think in general, right, doing a lab build, starting a lab, building a group, is challenging. It's sort of in the good ways and also the bad ways. There's a lot of things that are complicated that you didn't ever expect, as I'm sure you both know, and you know, sometimes you don't know if you're the rule or the exception. Like, is it always hard to order things sometimes? Or, how do you pick people to be in the lab? And what are the right things to ask? Sure, I'm telling you a story of the projects we picked, but there were other things we didn't do, right? Or there are things that we're going to plan to do that may or may not be as interesting. And so it's very interesting and can be complicated to know when you start up. Am I doing it right? Should I be doing something different? And there's lots of people here. Other people have started their labs, but you're very busy. Everyone's very busy, and it's kind of like, should I be asking people these questions, like whatever? And so I think it's been very nice to kind of have a person who is going through a different but parallel track, right? So much more. She's in engineering. She's in an engineering department, and she knows a significantly larger amount of math than I do. These distinguish the intelligence between us very obviously, but it's nice to kind of see the similarities and realize that there are lots of things that are generally complicated, and you just kind of got to work through them, and also think through, how quickly do you hire people, and how do you think about a thesis project for someone, and these sorts of things that are kind of topic agnostic but very relevant to building these things out. So it's kind of nice to have a person who's bought in into the effort, but also has their own experience to cross reference it with.
Yeah, it's so cool that you guys are starting something new at the same time, and it's interesting to hear how your experiences are different but the same, and I'm sure you guys must feed off each other. So bonus there. And also, yeah, thanks for sharing all that you shared about kind of the newness of a whole scientific field, and how you came into it and balancing the different choices and priorities. I mean, what I'll say I take away from all that is just the sense of commitment and conviction that anything new takes, a marriage, a lab, glycoRNA, and just dedication to moving forward, right? And addressing the problems that are right in front of you, whether that's, making new tools or, just being there for your partner and spouse. Thank you for sharing all across the board. Before we let you go, we got a couple kind of peripheral questions for you. The first one is, if you were not a scientist, what would you be?
It's a good question. I think that I would probably be an architect. Since I was very young, I very much liked drawing shapes and even structures that people could like live in, or I might live in one day, or work in. I've always found three-dimensional forms very interesting. I did a lot of pottery when I was younger, in high school and middle school, and I think I like imagining physical spaces in forms that are relevant to natural shapes, but use human-made materials. I like a lot of concrete, steel, glass things, but in otherwise not square configurations. So I'd probably think of building things like that.
Wow. I mean, I see the parallel right away. I'm sure you must too, but structural biology much? I remember when I first took my orgo class, it was such a revelation because I hate chemistry. But when I got in that, I was like, wow, this is cool because it's just a different way of thinking of it. I would picture the reactions, and that was so instrumental to me, not only understanding, but appreciating the beauty of biology. And I could see how, your what you said earlier about the RNA not just being a template, that being the kind of anomaly. And RNAs are structures, right? They're forms, and they bind and have activity. And now all I can picture is like furniture that's like folding and moving. It sounds like a horror movie. I got to put that away. Final questions. A few fill in the blanks here. When I'm not conducting research, I am.
I would say that I'm enjoying sci-fi. I think a lot of sci-fi. Although, I talk to Ritu about this a lot. Always prefer we would always prefer sci-fi that is less dystopian. Make the technology fun and exciting to live with. That seems also great. I'd watch a movie about that.
Anything specific that we can latch onto? Arun and I are total geeks about the sci-fi, so we'd love to chat after the show. But give a little kernel to the listeners, will you?
I mean, things that I've liked recently. Altered Carbon, I thought, was a very interesting world build. I just started watching Three Body Problem, although I also need to read the books. That sounds great. And even things. Severance was a very interesting concept, using some technology and things. That's again dystopian, but very interesting to think through these scenarios. The last one I'll mention, of course, is Black Mirror. Incredible imagination around a singular technology and how it impacts society. Of course, each of those, they find the worst way it could impact us, and then explore that, which is sometimes depressing, but it actually makes you think about what could you make and use eventually, and how will we interact with all of these features of technology.
Absolutely. 10 out of 10 on all those, except, I mean, come on, dystopian. Every single one of those will put you in the tank. Last thing I'd say is For All Mankind, for all you out there, if you want something inspirational, that's some real.
Halfway through that. I'm halfway through that. Yeah.
Solid. I just finished the final season. It's amazing. Nice. We could talk about that for hours. Let's get back to the show, shall we? Finally, a couple more. If I could have one superpower, it would be.
I would say teleportation, but to note, not constrained to Earth. I feel like a lot of people imagine it only on Earth, but it should be able to go anywhere. I would say. So
I love more than anything you and Ritu together, because I think we asked her a similar question, and she had this great answer of being able to like Ant-Man it and see all the molecules, like actually get down to the atomic level. And between that and teleportation, you two would make quite a terrifying duo.
Yeah.
I can't start the day without.
I'll going with an easy one. I'll say coffee for this one. But to make a point, only cold brew. I drink cold brew throughout the year. No temperature changes that outcome. It's always cold brew.
I will accept that answer, but the correct answer is actually to gaze at your spouse for at least a minute and just take it.
Okay, I'll practice that. I'll practice saying that. Yeah.
Back to the drawing board. No, just messing with you. Those are all amazing answers, and we really could continue this conversation for much longer, but we got to stop there. Again, Ryan, thank you so much for sharing all these insights. Really got us thinking. It's so great to have an innovator at the beginning of a career that looks like it's the end of most successful scientist career. You nailed it already, and now you're kind of just fleshing out all the pieces, and I can't wait to see how that picture ends up looking. Thanks for all your insights and thoughts, and we'll talk to you again on this show, I'm sure.
Awesome. It's been great to talk to you both.
All right, y'all. that brings us to the end of our show. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and Roundup papers. You can also reach out to us on X at STEMCELL Podcast or by email at info@stemcellpodcast.com with feedback or to suggest guests.